So welcome to the IBSS seminar series. Thank you for joining us. If you are just tuning in now, please remember to mute yourself and turn off your camera. And I'll introduce this week's speaker. So Dr. Molly Womack is a postdoc in my research group in herpetology at the Academy, but she is starting her own lab in a couple of months at Utah State University. And so I first met Molly at the herpetology conference one summer, and we really hit it off. And so I was thrilled when she reached out to me about applying for the National Science Foundation special postdoctoral research program with an emphasis on museum collections. So Molly had done a lot of work during her PhD, and even though she was at an institution, Colorado State, that doesn't have a large research collection, I think they just have a teaching collection, she'd found ways throughout her PhD research to use museum collections and visit museums to incorporate collections into her work. And so this postdoc program was a perfect fit for her because it was going to build on that interest. And so most of her PhD research was focusing on the evolution of ear loss in toads, which she will talk about a little bit today. So both the diversity of cases in which this happens and also some of the consequences of ear loss. And for her postdoc project, she was interested in broadening out, not just looking at toads, but looking across all frogs to look at morphological evolution and leveraging museum collections and also new advances in CT scanning technologies. And so that was her first postdoc. And then her second postdoc, she got the Smithsonian Institution's Buck Postdoctoral Fellowship and also the Secretary Scholar Award, which is just given to one postdoc across the entire institution every year, so a very prestigious postdoc. And to continue her work looking at ear loss, not only in amphibians, but also broadening out to reptiles, which have also lost ears many times. And so Molly is based in Berkeley, and she has been, because she was co-advised for her first postdoc. So she's actually semi-local. And so even though I moved from the Smithsonian to the Academy, she's been able to spend some time at the Academy and the NMNH in DC and UC Berkeley. So she's been traveling quite a bit over the last few years. And fortunately, we are actually in the same place most of the time, although spending a lot of time on Skype and Zoom these days, even though we're in the same city. So anyway, I'll let Molly take it away, but that's sort of her academic background, and keep an eye out for her starting her new lab at Utah State. And if you know any students or postdocs that are interested in the research that she's talking about today, please send them her way as she builds her lab.
Great. Thanks, Raina. Okay, so as Raina mentioned, I'm going to talk to you today about my work mostly on skeletal evolution of frogs and toads. And I'm currently finishing up that Buck postdoctoral fellow, but as Raina also mentioned, I'll be starting my lab August 1st at Utah State University. And for those of you who aren't familiar with Utah State University, if I pull up something most people on this chat probably are familiar with, INAT records, all INAT records of frogs in the US. My future lab is right here in this gaping hole of frog diversity, which is pretty funny given that frog diversity is mostly what my lab is focused on. So my lab in general and what my work has focused on in the past is documenting phenotypic diversity we see in the natural world, and then asking two big questions about that diversity. And mostly I've worked on frogs for reasons I'll explain throughout the talk, mainly historical contingency, but also because I think they're so useful for studying evolution. And the first question I want to know about the diversity we see is how is it generated? And so for that, I'm talking about how does variation in genotypes lead to variation in phenotypes we see? How does this phenotypic variation reflect where animals are in the world? How have their behaviors and climates and experiences selected for these varying phenotypes? And then also, what can we learn about the relationships among species to understand how phylogenetic constraints might be constraining evolution and the phenotypes we see in nature. So this is basically a question about the evolutionary process. What can we learn about evolution from the diversity we see? And then the second big question that Raina also alluded to in her introduction that I like to ask is why does it matter going forward? So why does this phenotypic diversity matter for any given species? And so will this matter for them going forward as far as their ability to diversify or their ability to thrive in a changing environment? And so I've been focused mainly on skeletal evolution in frogs for asking these questions. But today I'm actually going to talk to you specifically about one single frog bone, one tiny little bone, the frog middle ear, highlighted here in orange on this toad skull. And the reason I want to talk about frog ears is not only because they show interesting diversity, but because I think this one tiny little bone actually has a whole lot to teach us about the process of evolution and highlights an understudied important aspect of the evolutionary process. And then the other thing this project does is incorporates museum collections in a ton of different ways. So first, before I answer all these lofty questions about diversity in frog ears, I want to introduce you to the frog and toad ear. So for those of you who haven't been studying ears or thinking about them, frogs have ears. And if you pick up pretty much any North American species, you see this circle here right behind the eye. And the circle is actually just a thin tympanic membrane shown here by this 3D reconstruction. This thin tympanic membrane is similar to the one you have. It's just that frogs don't have this kind of sticking out bit that we have from our ears, the pinna. So they have their thin tympanic membrane exposed, surrounded by this cartilaginous ring shown here in blue for support. These structures that you can see from the outside of the frog connect to the inner ear within the skull via this mostly bony columella or stapes. And the way these all function is that a sound like a calling toad hits this thin tympanic membrane, causes it to vibrate. This vibration then moves that middle ear columella and sends those vibrations to the inner ear fluids, where those motions, those fluids are sensed by hair cells. These hair cells send a signal to the brain that the frog or toad has sensed a sound. And what's really interesting about frog and toad ears is that a lot of species are earless. So they lack all these structures that bring outside noise to the inner ear. And this is really interesting, not only because frogs and toads are well known for generating calls and using calling for acoustic communication, but also because this is how pretty much every land-living tetrapod gets sound from the outside environment to the inner ear. It's how you're listening to me now. It's how your dog hears. It's how your cat hears. And frogs are really kind of superb in the number of times that they have lost these seemingly important structures. So to visualize this, here I'm showing you a tree of life of frogs. This one contains a few thousand species of the more than 7,000 currently known. And on the edges here, we have colors. The orange represents species that are known to have a middle ear, and the blue species represent individual species that are known to not have a middle ear. And what's really cool about this is you can see just at a glance that there are a lot of earless species all over this tree. So earlessness has seemingly evolved at least 38 times, and we know there are a lot of key independent lineages that are not represented on this tree, so it's likely many more losses have occurred. And the other interesting thing that we can see on this tree is not just that ear loss happens a lot, but that it's not randomly distributed. We see these kind of clusters of ear loss in particular areas. And so this, what I'm highlighting here in red, is the family Bufonidae. So these are the true toads. So these are the whole clade of animals that you actually should call a toad. And in this family, we have 12 to 17 changes in the ear. The number of transitions depend on the likelihood of regains, but basically we have a whole lot of loss happening and way more than any other family. So other families, losses happen maybe just once or twice and in a few species. Here we have many more species than in any other family, and also many more independent losses. So this started the question that I would then try to answer for 6 years of my PhD. And it's a relatively simple question, seemingly, which is that just why is this happening? Why are these middle ears lost so often? And so you could think of a number of reasons. Many people had thought of many reasons that the middle ear might be lost related to all these hypotheses I've put up here. And so I'm just going to walk you through what I've found and what we know about the possible reasons for this ear loss. So the most obvious explanation for species converging on similar phenotypes is always habitat. So is there something in the environment of these earless species that's selecting for ear loss? And so if we look geographically, here I've plotted where earless toads occur. So every country that has an earless toad species has a dot. The bigger that dot, the more earless species that country has. And what you can see is there are a lot of countries that have earless species. And so we don't see any kind of limit in geographic representation here, although there aren't that many in North America, Europe, or Australia. If we expanded this to earless frogs, not just earless toads, we would see a lot of earless species in these regions as well. So earlessness is kind of all over the world. But what's really interesting is that it doesn't show any relationship with habitat. So we see earless species in deserts, we see them next to raging waterfalls, we see them living in low montane streams, we see them in high montane forests. So there's really no habitat correlation with ear loss. And when we look at a more micro scale, we don't see any difference between the ways that eared and earless species are interacting with their environment either. So a similar proportion of eared and earless species are aquatic or semi-aquatic, spending most of their time in water. A similar proportion are burrowers or semi-burrowers, often burrowing for part or big chunks of the year underground. Then there's a similar proportion of terrestrial species and a similar proportion of arboreal species found mostly in trees. So there's no, on any level, correlation with the environment or the habitats or the microhabitats that these species live in and the lack of a middle ear. And this is really in contrast to other clades where we see ear loss, such as lizards. In lizards, it's usually burrowers that lack a middle ear, and they're really covering up that tympanic membrane is usually what they're doing to limit grit and sand getting into that ear. So it's very kind of understandable why this is happening and highly correlated with their microhabitat. Okay, so for frogs, we can't explain this convergent ear loss by habitat. Another possibility that we thought of is it could be a size constraint. Frogs get really small. They're some of the smallest tetrapods. In fact, they hold the title for the smallest tetrapod on Earth currently. And so when you get that small, sometimes you lose bones simply because you lack space. And with the ears being right next to the brain and the skull, you could see potentially losing an ear to kind of due to constrained size in the skull. So if we look at body size across frogs and toads, so this is including all frogs and toads, not just the family Bufonidae, and you look at eared and earless species, you do find a correlation when accounting for phylogenetic relationships. Earless species are smaller than eared species on average. But what's interesting about the data is what you don't see are really big earless species. But what you do see are really, really small eared species. In fact, some of these smallest species on Earth have a middle ear. So it doesn't seem to explain ear loss because you can be incredibly small and still have an ear. Okay, so size has an interesting relationship with ear loss but can't explain it. When we see changes in the skull, another go-to hypothesis is always maybe there is some shift in diet that's selecting for changes in the skull. Frogs and toads, although we were chatting a little bit before we started here about variation in frog diet, in general, most species are just eating whatever they can fit in their mouth. And so we don't see anything like we see in snakes where you have extreme modifications in the middle ear to expand your gape and be able to eat larger prey. I have looked in a number of closely eared and earless species to compare muscle morphology, especially these large feeding muscles that, as you can see, are interacting and developing right alongside the middle ear. But I don't see any differences between eared and earless species to indicate that there's any sort of shift in jaw strength or the ability to clamp down on prey between ear and earless species. And this is actually work I'm expanding in this postdoc at the Smithsonian in comparison with lizards and snakes, but as you might expect, it's a little bit delayed from COVID. But there's no real indication as of right now that diet plays any role in middle ear loss. And so one of the main hypotheses for middle ear loss has to do with sensory system evolution. So obviously the middle ear is directly tied to the sensory capabilities of the frog. So maybe there's some explanation in how those are varying. And probably the most popular hypothesis for ear loss, or at least the most discussed in the literature when I started my PhD, was, do earless frogs have alternative hearing mechanisms? So there was this idea that earless frogs might be able to hear even though they lose their middle ear. And there were a number of proposed extratympanic hearing pathways, so these other ways that frogs and toads are getting sound from the outside environment to the inner ear. I've kind of roughly diagrammed them here, but they all involve some part of the frog vibrating, and then those vibrations being transferred through various tissues and bones to the inner ear. But the experimental evidence for this was pretty rough and only in a couple of species. So to get at this question, I focused in on toads. Reason being, we could test the hearing of closely related eared and earless species pairs. So I ended up testing the hearing of 10 toad species in total, 4 earless and 6 eared. And I was able to capture not only multiple independent losses of the middle ear, but also variation in lots of other things we know affect hearing, like body size. So how do you test the hearing of a frog or toad? We did these auditory brainstem recordings in the field. And so these are a really easy way to get hearing tests on pretty much any small animal. And it's similar to what they do, or at least what they used to do up until recently with newborn babies. So when a newborn baby's born, you put a patch on the head and right by the ear, And then you play a sound, and the patches record electrical signals that are running from the inner ear to the brain, signaling that the baby can hear sound and there's no problem with the baby's hearing. We do a similar thing, but we use these small electrodes that go just underneath the skin of the frog. And this allows us to temporarily immobilize the frog, put it in this makeshift audio chamber that you can take into the field with a speaker on one end to play tones, your frog or toad on the other end, and then you can play sounds at increasing intensities to the frog until those electrodes sense a signal running from the inner ear to the brain that the frog hears it. So what sort of data comes out of these? This is an example of the hearing sensitivity curve of one species. So this is showing you how this species can hear. And so on the x-axis, we have tones running from 200 hertz, which you just heard there. Maybe you heard to 4,000 hertz at the high end. And so what this really covers is the normal hearing range of most frogs and toads. And then on the y-axis, we have threshold in dBSPL. So this is basically how loud I had to play the sound for the toad to hear it. So we have good hearing, low on this y-axis, and poor hearing, high on this y-axis. So if we look at this example going through this one species, they have pretty good hearing at lower frequencies. Right around here is the level of normal conversation. So if it was just you and a friend in a room talking at very normal levels, that's about the decibel level this line is at, and the toad could easily hear that. But then the hearing curve really shoots up, and we're seeing poor hearing in this species, and I'm having to play the sound about as loud as a lawnmower before the toad can hear it. So that's pretty bad hearing. So now if I throw all the data at you, this is what we see, kind of a mess of blue and orange lines. Again, and throughout the talk, eared species are in orange, earless species in blue. And what I want to do is focus your attention to the left portion of this graph. So to the left of this line, what you see is a lot of the curves are overlapping, indicating that all species, regardless of whether they have an ear or not, have similar and pretty good hearing. But then if you go to the right of this line, you see the variation really start to expand in species hearing capabilities. And what you hopefully also notice is that there's a lot of blue lines high on this graph with poor hearing, and a lot of orange lines lower on this graph indicating better hearing. And indeed, that's what we find. Every frequency above 900 hertz that we tested, there's a difference between eared and earless species, with earless species having poor hearing. And this not only holds as an overall pattern in the data, it also holds when you look at really similar looking, closely related eared and earless species pairs. So what we found, although maybe not entirely surprising, but excitingly, is that hearing is not retained in these earless species. It does matter whether or not you have an ear, how well you can hear. So it's a 16 to 25 decibel hearing difference above 900 hertz. That's kind of the amount of hearing change between an eared and an earless species. But of course, why does it matter? And specifically, why might this matter for social communication, since we know social communication, acoustic communication is so important in frogs and toads. So if an earless species can hear some sound from X distance, an eared species can hear that same sound, whether it be predator, prey, or a calling conspecific, from 4 to 16 times the distance. And to me, that's going to make a really big impact on whether or not, on mate choice, whether or not you're able to even hear or understand the presence of a mate or a conspecific or a predator in the area. And I should also mention here, an interesting note about earless species is that they are still calling. So earless species still call, and in fact, they call often at high frequencies. So exactly at these frequencies where we see this worse hearing in earless species, those are the frequencies they are calling at. So it seems like the lack of an ear is maladaptive. Okay, so they're not getting by without an ear, at least not hearing-wise. So high frequency hearing, the ear is important for that. Maybe they're compensating with alternative sensory modes. So maybe earless species can't hear as well, but doesn't matter because they're using some other sensory modality. And so to look at this, I actually wanted to focus on vibration sensitivity. So do earless species better sense vibration? And the reason I wanted to look at vibration is, one, we knew that that frogs and toads were some of the best tetrapods at sensing vibration that have ever been tested. This was discovered in the 70s, but hadn't really been followed up on. And in relation to earlessness, vibration is pretty interesting because, one, those vibrations are being sensed by the same hair cells that earless species and eared species both have. And they also, the mechanism for bringing vibrations into the inner ear is this proposed pathway shown in red here, the upshot of which is that it comes through the skeleton, mainly that's thought by the forelimbs, goes through a muscle, and then this cartilaginous disc by the inner ear vibrates and sends that signal into the inner ear. And this cartilaginous disc is what I'm showing to this in highlighted in red here on the right photo. And what's cool about this cartilaginous disc is it's actually almost encompassed and wrapped by the middle ear bone in eared species. And so the thought was that if you remove that middle ear bone that's almost kind of hugging that disc, you might free it up and allow it to move, have kind of greater mobility, and therefore increase the signal of vibration in earless species. So this was the hypothesis. And so to test whether earless species might have enhanced vibration sensitivity, we did more ABRs. This time, instead of playing tones in a box to the species, we did ABRs where we played them vibrations on a platform, but the same general process. So I'm going to jump right to the data this time. So here's all the species data this time all at once, those 10 same species where I did the hearing tests. And what this is showing is, again, on the x-axis, we have frequencies. So I'm running from 200 hertz again, but this time stopping at 900 hertz because higher frequencies don't travel as far through substrates, so we weren't as interested in those. And then on the y-axis, we saw thresholds. So lower is better vibration sensitivity, higher is poor vibration sensitivity, and this is an acceleration. So to give you a little bit of a touchstone here, an iPhone, or at least an iPhone 4, buzzes at about this threshold. And so what this difference between this iPhone indicator and the toad sensitivity shows you is that toads are incredibly sensitive to iPhone vibrations. And your fingertip sensitivity is tracking this white line. So you are also incredibly sensitive to your iPhone vibrating, but toads are much, much more sensitive than you to vibrations. And so a good kind of fun example for anyone out here on the West Coast is that how your iPhone vibrating feels to you, to that toad, it feels like a 6G earthquake. But what you don't see on this graph is a lot of difference between these orange and blue lines. So eared and earless species are both sensing vibration sensitivity really well. There's really no differences. In fact, I couldn't even put the error bars on this plot because you wouldn't be able to see the different lines at all. So earless species are not more sensitive to vibration. this doesn't seem to be a compensatory mechanism for ear loss, but it has led to a ton of follow-up studies for how frogs and toads might be using vibration. And this is a particularly fun collaboration of a few I have in this arena with Beta Ruida, who works on these beautiful harlequin frogs shown here on the left. So these Atelopus are found on these leaves at night, kind of perched, just like this photograph shows. And in a couple of the species Beta has been observing for many, many years, you always find them in these kind of upright, kind of alert-looking positions in the evening, and you can actually go to leaves and tickle the leaves, and they will orient to the vibrations that you're creating and then try to eat the probe. And so he's doing all these playbacks, and we've done vibration sensitivity tests in his species to kind of show that they might be using this for prey capture of insects, which is really cool. So although we found really interesting results about sensory system evolution in relation to earlessness, we didn't find anything that really explains why these frogs and toads might be losing their ears. And so the final hypotheses that we wanted to test about why frogs and toads might be losing their ears all had to do with development. And again, here we had two hypotheses. One was that middle ears might be lost due to developmental pleiotropy within the skull. And so the idea here is similar to what's been seen in eyeless cavefish. So fish that inhabit caves often have a suite of characters that are convergent. As you can see from these photos, lack of pigmentation is a big one, but also degeneration and sometimes complete lack of the eye is another common cavefish convergent trait. And for the longest time, the kind of narrative as to why eye loss was occurring was there was relaxed selection because you can't see in these dark environments, so the eye isn't very useful. And then the eye is very costly to develop, and it's costly to maintain. And so these two kind of forces, the relaxed selection and then the costliness of the eye together, lead to it being degenerated over time. But what's really cool is developmental work has actually shown that these eyeless cavefish have more taste buds and enhanced taste sensitivity compared to their non-cave counterparts. And what's even cooler is the development of these enhanced taste buds actually is linked to the degeneration of the eye. There's this genetic and developmental pleiotropy such that if you want to enhance these taste buds, you actually end up degenerating the eye. And so it's actually this developmental trade-off that plays a big role in eye loss in cave species. And so we were wondering if something similar was happening with ear loss in toad skulls. So maybe in frogs and toads, you have a similar trade-off where you're losing the ear, but there's something genetic or developmentally linked in the skull that you're gaining as a trade-off. And so here I'm showing you a developing tadpole skull. And so everything that's colored on the skull with blue or yellow or orange is made from three neural crest streams. And these three neural crest streams make up the different parts of the tympanic middle ear. And so all this illustrates is that the middle ear is developmentally tied to many aspects of the developing skull. So to test whether these kind of developmental trade-off or developer genetic pleiotropy might be contributing to ear loss, I dove into the National Museum of Natural History collection, and they graciously loaned me a ton of species so that I could microCT scan their skulls and compare to see if there were any convergent structural changes aside from the loss of the middle ear. And so here's our sampling. We have more than 50 species here with at least 7 ear loss, independent ear loss events. And so what we wanted to know is there some consistent change, some consistent difference between eared and earless skulls? And what I'm showing you right now is an overlay from the top down of a toad skull And this is showing you the maximum difference between eared and earless skulls. Kind of if you make eared and earless skulls as different as you possibly can across these species, this is your big difference. And hopefully what you see is that these lines are nearly overlapping. So earless species are a little bit thinner in their skull and ever so slightly longer, but we're talking about, I mean, millimeters of difference here. And so if we look at this in a different way on a PCA plot for anyone who is more used to looking at data that way, what you see is a lot of blue and orange dots in similar places on this graph, which indicates those skulls are very similar. And we can overlay the phylogeny and show that really there's no difference between eared and earless species in skull shape. And the other thing I checked for was whether earless skulls might be missing other skull bones. And I paid special attention to these other bones that develop around the same time as the middle ear and are lost in many other frog species. But none of the earless species were missing these bones. They were present in every single species except one was missing one of these bones. So ears seem to be lost in isolation in these convergent ear losses, at least in toads. So we have no evidence of some trade-offs, at least with skull structures. We don't see any evidence for developmental pleiotropy in the system. But there was kind of one last hypothesis that had been brought up again and again in literature, which was that maybe middle ears are lost due to changes in developmental timing or rate. And this is termed heterochrony, and it's a really common way for evolution to occur in amphibians in particular. And so the reason people thought that it might be changes in developmental rate or timing is because middle ears develop really late. So most frogs and toads have this tadpole phase where they're swimming around in water. Their skull and almost their entire body is all cartilage, no bone, very little bone. Then they go through this crazy metamorphosis where they resorb their tail, they grow their limbs, and they pretty much rearrange everything in their skull and body. And they often also transition at this point to living on land. And it's during this kind of insane time of transition that the middle ear just starts to form. And in many species, it's known that the middle ear actually doesn't complete formation until well into the juvenile stage, so months after metamorphosis. And this is in contrast to most aspects of the skull and the body in general, which are pretty well developed and in place a couple days after metamorphosis. So perhaps this really late forming delay structure would be incredibly sensitive to delays or slows in development rate. And so the idea being that maybe if earless species are developing more slowly, this late forming bone is just kind of pushed off the developmental cliff. But I don't have to tell many people at the ACAD how challenging it can be to rear especially many different types of species. So to do this comparison about are there differences in developmental rate between eared and earless species, we actually looked to an indicator of development rate, which would be genome size. So in amphibians, there's been a lot of work to show, and actually across animals in general, that larger genomes leads to longer cell proliferation rates and longer developmental times. And so to measure genome size, genome size is known for a lot of eared species, but very few earless species, I again went back to collections. And this time, again, at the National Museum of Natural History, I extracted heart cells from fixed specimens. And then I was able to do this Fulgen stain, which essentially just stains DNA content. This allows you to image the kind of nuclear densities of across species. And this allows you to get an estimation of genome size as long as you line it up with some species that you know genome size for. And so what we were able to do with this data is, one, show at kind of the largest macroevolutionary scale at that time that, in fact, genome size and the time spent as a tadpole does have a positive correlation. So larger genomes does mean longer development in frogs and toads, and this is across many families. And as you can see, there are very few earless species for which we know development rate, but we do know it for quite a few eared species. So kind of armed with this positive correlation, which I should note here is mainly driven by the lack of species with large genomes. Large genomed species cannot develop fast. You can develop very slow if you have a small genome, but you can't develop fast if you have a large genome. But armed with this kind of positive correlation between development rate and genome size, we then looked and compared eared and earless genome sizes across the phylogeny. So here's our final sampling of 206 species with known middle ear status. And what we find is that earless species indeed, on average, are larger than you would expect compared to their eared counterparts. So you can clearly see, again, this is not a real explanatory relationship. We don't have all species that are earless having extremely large genomes, but you do see these somewhat consistent shifts towards larger genomes compared to closely related species as you transition to the earless phenotype. Okay, so we did see this kind of prediction bear out that earless species might be developing longer, and maybe this has something to do with the evolution of earlessness. We had one other indicator that development might be important, and that's going back to this idea that true toads, this one family, has a lot of ear loss, way more than any other family. And it had been noted in some studies done in the 70s and 80s that they also developed their ears really slowly. So in Rhonopipians, a different family takes only a day or two to go from no middle ear to fully developed middle ear. In another group, in hylids, takes up to 60 days, at least in the one known and well-studied species, to go from no ear to fully developed ear. But in toads, in some species, it was taking almost a full year after metamorphosis for that middle ear to be fully formed and functional. And we, in fact, showed that across development, until that process isn't complete, until the middle ear is completely ossified and fully formed, it doesn't function at all. So it's really unhelpful up into 1 year past metamorphosis in these toads. So extremely delayed, and maybe this has something to do with the fact, or maybe the fact that it takes so long for toads to develop their ear has something to do with the fact that they lose it so often. So then we did the really hard thing to try to actually get at this more directly, and we reared eared and earless species of toads. So two closely related eared and earless species pairs, one in the genus Rhinella, one in the genus Atelopus. And we would not have been able to do this without these two centers for sustainability, so Centro Hambatu in Ecuador, and this other conservation Center for Conservation, Development, and Sustainability in Peru. So in collaboration with these centers, we were able to rear these toad species very close to where they're naturally found. And then at this point, a postdoc, Jennifer Stanowski, joined the project and did amazing work comparing the development of not only the ear, but also surrounding structures across these developmental series. And what she was able to find was that both eared and earless species start developing their ear at metamorphosis right at the same time. So regardless of whether they have ears as an adult, all frogs, or at least these toads that we've studied in detail, start to develop that middle ear. And it's just in the earless species, at some point, and it varies between the species, this process halts and what has grown out of the ear resorbs. And even more interestingly, in some cases, it resorbs completely. There's no evidence at all in adults. But sometimes even of individuals of the same population where you see no evidence of the ear in adults, you'll find individuals where there's chunks of middle ear still there that haven't disappeared. And this is really cool because this is noted in things like legless lizards start to develop their limbs and then resorb them. Whale pelvises, it's been known for a long time, start to develop some of these structures and then resorb them to varying degrees. So this seems to be a common developmental rule that a lot of structures that we think of as lost are not lost at these early stages. And so we're following this up to better understand the genetic and developmental processes at play when, why do these structures stop forming? And then what's the process of degenerating that tissue that's already there? So we have lots of tissue and RNA later from these developmental series. And we have some candidate genes that we're excited to explore with that. So I received a DDig during my PhD, and again, museums, including the ACAD, were generous with their tissues, and we took tissues from the Museum of Vertebrate Zoology, the ACAD, and the National Museum of Natural History, and we're able to build a phylogeny that included a bunch of earless species that weren't currently on toad phylogenies. So that's this phylogeny to the left here. We were able to add earless species, including new ear loss events. And this was in collaboration with the Center for Anchored Phylogenomics. But the other thing we did was we sequenced a ton of candidate genes that we know or we thought were involved in ear development based on studies in Xenopus, but also studies that show ear genes being conserved across all tetrapods. And so we ended up with about 30 genes that we got really good sequences back. And these genes are really conserved across species because they're not only forming the ear, they're often involved in development of many structures in the skull and in the body in general. And so these 30 genes in general, they showed incredibly low DNDS ratios, so non-synonymous to synonymous mutations. And all this really means is that all these orange dots that you see here are close to the 0 line, indicating that there are very few changes. These are under high purifying selection, or not purifying, they're highly conserved across species. But for the 6 genes I'm showing here, we actually found relaxed purifying selection in the earless species. So earless species for these 6 genes actually showed a little bit more mutation than you would expect compared to the eared species. And a few of these differences are quite small and somewhat inconsistent with the different ear loss events, but TBX2 is really popping out as an outlier here and consistently shows elevated mutation rates in earless species. And so that's something we're really hoping to follow up with these genetic studies that are a little bit, again, on hold for COVID. Okay, so what have we learned about frog ears? So the first thing we learned was that ear loss is rampant across the frog tree of life. But getting at these kind of bigger questions of how it's generated, we see that, and I think I'm coming up on time here. Okay, still have time, good. So we don't see a strong association between ear loss and the environment or the habitat or anything these frogs are doing. So that was really the surprising thing, and this is the really interesting thing about ear loss, is it's happening a lot and we don't see any selection pressure for it. We don't see any easy explanation for why these might be evolving again and again. And so this leaves us with kind of an unknown explanation, or at least no kind of universal selection pressure for earlessness. But what we do see are these really interesting indicators that development might be playing a strong role in predisposing ears to be lost. So the kind of late formation of this trait might be contributing to its loss across the phylogeny. So then thinking about this other question of why does it matter? Why does it matter whether a frog or a toad has an ear or not? Well, there's kind of the obvious answer, which is it matters for their hearing. It matters how they can communicate and sense the world around them, which seems potentially consequential. But also something interesting is that, you see very tiny clades of ear loss. You don't really see earless radiations. You don't see a ton of earless species. So even though individual clades, these small pockets of earless clades, seem to be doing fine, some cases those species seem to be thriving. It does seem like potentially it's hard to diversify once you evolve the lack of a middle ear. Once you lose that middle ear, it might be a little bit of an evolutionary dead end, but it's really hard to quantify this in any meaningful way with only extant species knowledge. And so not something I'm going to say is for sure happening, but something I think about and something I think is really interesting. Okay. What I think is actually the most interesting about earless frogs is that they shine this light on this understudied process, which is developmental bias. So they really are kind of a candidate example for how an organism develops can affect how it can evolve. And this is something that has been appreciated for a long time, but finding examples, clear-cut examples of this in nature is pretty difficult, and it's pretty difficult to show that it's really this developmental bias and not some selection pressure. And even within earless frogs, this is an example that shows that this influence of development can really have downstream consequences, in this case on communication and diversification. And so to hammer this idea home of developmental bias, there's a lot of studies focused on how variation in genotype leads to variation in phenotype. But development is this really important step in that process. This translation from genotype to phenotype happens through development, and developmental bias shows that in this developmental process, it actually has the ability to kind of shape what phenotypes are possible, or at least make some phenotypes more likely to evolve, such as in this toy example, these blues and greens, then make other phenotypes less likely to evolve, such as these red and orange phenotypes here. And so this is kind of what we think is going on with earless frogs and toads. Because the ear develops very late, because of how that ear develops, it's predisposed to popping up as a phenotype. And this has led us to question, are late forming structures more evolutionary labile in general? And so this is something that we know is potentially true in frogs. So these late developing middle ears are easily lost. These other late forming skull bones are lost in other clades. This really late developing extra digit, but it's not really a digit. This is actually a ACAD specimen shown here in the center. This is often lost and changed a lot over the frog tree of life. It's also something we see in other clades. So digits in mammals, the last ones to form are the first ones to be lost. Late forming appendages like tails are often lost across mammals as well. So I wanted to investigate this across the rest of the skeleton during my postdoc. So harnessing museum collections and microCT, bringing all the skeletal diversity of frogs and toads together, kind of making a heat map of what's changing a whole lot in the frog skeleton over evolutionary time and what's changing very little. And how does that relate to when those different structures form? So the idea that maybe early forming traits, early forming skeletal bones don't change as much as late forming skeletal bones. And as I end here, I'll just point to some really great work. This is a horrible screenshot, but this is a paper just accepted to evolution out of the Goswami lab. Beautiful work by Carlo Bordua, who shows that this relationship is true for skull bones. There's higher variation in later developing bones. There's some work that is about to be published in Evolution by a student I mentored, Natasha Stepanova, who showed that the early forming limb bones of frogs have lower evolutionary rates than these later forming limb bones shown in these lighter colors here. And I've been investigating this again across the skeleton. So for anyone interested in other frog bones, get ready for a lot of work coming out from these amazing students, both high school, undergraduate, and graduate students. And so with the little bit of time I have here, just check how much. Okay, so with a little bit of time here, I'll just wrap up by saying I think frogs are particularly useful for studying evolutionary processes because they're found all over the world. I usually say except Antarctica, but recent articles showed, at least in the past, they have been on Antarctica. They do all sorts of crazy things, and they've convergently transitioned to living many different lifestyles, which makes them really helpful for looking at relationships between what an animal does and how it looks. Frogs are also pretty interesting genetically. I showed a little bit today. There are differences in genome size, but they have all sorts of other cool things going on in their genomes. They vary in development. I showed today differences in development rate that are known across frogs and toads. But I didn't even mention the crazy transitions from having that tadpole phase to not having a tadpole phase and developing fully within an egg. Those transitions have happened multiple times across frogs. And so all of this together really makes frogs a powerful system for studying how all these different things evolved and how they shape the diversity we see in this clade. And beyond what kind of frogs can teach us about evolution, I think they're a particularly important clade to be studying because understanding their diversity not only allows us to document what we currently have here on this planet, but also will help us conserve it going forward. And I'm going to keep doing this type of research using, of course, museum specimens, which are great not only for the phenotypes they allow us to document at a comparative scale, but frozen tissues, multi-stage and age data, all tied to observational data, location data, and time data. And this kind of ability to look through the past, I know I'm preaching to the choir here, but it really is a powerful tool for being able to predict how things are going to change and how animals will be able to adapt to future change. And I think also iNaturalist data is a whole new layer of this, and I'm really excited to see the ACAD is at the forefront of incorporating iNaturalist data not only into research, but into outreach and getting students involved in documenting diversity around them. And I'll be filling in some amphibian data gaps here over in Utah soon enough. And with that, I'll thank members of the postdoc labs I've been a part of, the Bell Lab and the Rosenblum Lab. The Hoek Lab is where I did most of this work, various collaborators, the museums, of course, and these funding sources. And I'll take any questions.
Great. So if you have a question, just type Q in the chat box, and then I'll call on you to ask your question. But yeah, thank you, Molly, for a great talk. Let's see. So Darrell has a question. Darrell, do you want to turn on your mic?
Sure. Great talk. That was super interesting. What pathways are those high DNDS genes involved in? And if you want to, can you speculate if there'll be pleiotropic effects of changes downstream from those loci that could lead to both some selected phenotype, since they're under selection, and a loss of ears?
Yeah, so the DNDS rates don't indicate that any of those are under selection by any means. They're well below that threshold, just that they're not as intense purifying selection. But I will say the only genes that are really known for the middle ear in most cases are genes that are highly pleiotropic, at least overall. And so I was really surprised to see any differences in these genes, just because it seems like if you mess with those genes, you're going to mess with a whole bunch of phenotypes. And as I showed in the skull data, we don't see these earless species looking particularly weird. And so I would be more, I think it's more likely we're going to see differences in kind of the timing of expression of these genes. So that's my guess. And so that way the ear is modified, but none of the other things are. And so it's quite possible that those elevated rates in the actual gene itself may not end up being important. I mean, I'm just kind of, I was surprised to see them.
Okay, thanks.
Yeah.
Great. Okay, so Ori, I don't know if I'm saying your name right.
It's Or.
Or,
hi,
okay.
this was incredible. I mean, it was so beautiful. My question is, you mentioned that earless species tend to make high-pitched vocalizations, and I'm wondering, in light of that, if you do actually want to study how they pick up, whether there's a difference in how they pick up vibrations at the higher frequency range.
Yeah, I knew much less about vibration when I went into this project, and we were under the, I mean, it is true, they don't translate as far across the ground. A high frequency vibration just isn't going to go very far, and so it's not that useful. But what we have since learned is that high frequency vibrations go very well through plants. And so now we actually think that those high frequencies may indeed be important. And one of the other follow-up studies I didn't mention is looking at agalicnus in tree frogs that do these tremulations. And we do see these higher frequencies. Not only are those frogs sensitive to the tremulations at higher frequencies, but they seem to be kind of tuned into the frequencies that this other researcher, Michael Caldwell, sees going through the plants. So yeah, I do wish I had actually done those higher frequencies way back then.
It's not too late. Thank you.
All right, Manuel, if you're still here. Okay, well, Manuel's question, oh, he can't talk. Okay, was when you talk about developmental bias, are you considering epigenetics or changes at the chromosomal level?
Yeah, to some degree, yes. Basically, in general, the developmental bias concept says it basically applies to anything that happens during early development that might affect what phenotypes arise. So that's everything like what I discussed, just kind of timing of when things are occurring. But epigenetics can fall into that as well, because certainly epigenetic changes could affect or predispose certain traits to change more than others, and in which case it's similar.
Great. Okay, well, thank you, Molly, so much for making time to chat with everyone. Oh, okay, there's another thank you. I was like, we have one more question. And thanks everyone for tuning in. And I don't know if Natalie has any announcements about upcoming seminars, but.
No, I'm good. We have a seminar next week, but that's in the announcements in the emails.
Great.
It'll be about citizen science. Thank you.
Thank you.